Hey folks, this is Mr. Ferrari. This is my second now in a couple series of quick videos to review some of the topics for this unit. If you missed the first one, that is my mitosis and meiosis review. I will reference a little bit of that in this one, but you don't need to watch the first to watch this one. But they, in the grand scheme of things, are fairly related. So with that being said, this one is going to be a quick review of both monohybrids and dihybrids. Hopefully the monohybrid review is pretty self-explanatory for you. It's a quick review. You might even skip through it. That's completely okay. What I've got written here, however, if should you stay, is I've got one trait here, and this trait is about heart rate. What I have written here is that represented by the dominant allele big R, the low heart rate is dominant to the recessive allele of a little r that is representing high heart rate. So low heart rate is dominant to high heart rate. Okay, again, because this is represented by the capital R, we can say that low heart rate is dominant to the recessive allele, the little r, of high heart rate. And I'm saying alleles here, right? These are not specific traits. These are the traits, but these are just alleles. These are what tell us what trait we may or may be showing or not. So let's take a look at that. Let's say that there's an individual who is heterozygous and they mate with another heterozygous individual. Remember that heterozygous means two different alleles. These are heterozygous individuals. They have two separate alleles. They have one dominant and one recessive. What this tells us is that both of these individuals, their phenotype is going to be the low heart rate, while their genotype might be heterozygous. Their genotype is heterozygous. Their phenotype is the low heart rate. This is their trait. This is the trait that we talk about. When we say, oh, someone has brown hair, or they've got a widow's peak, or they have freckles, this is their actual trait, the phenotype. Now, their phenotype is determined by their genotype, sure, but their genotype is made up of alleles, and those alleles are what give us our traits. They give us our phenotypes here. Okay, so this person, again, these two are heterozygous individuals. They have one big, one little, one dominant or recessive. That is their genotype. And so phenotypically, their trait is the low heart rate. And this is because in a dominant recessive relationship, if there is at least one dominant allele, then this individual would show the dominant trait. And so realistically, this person would also show the same phenotype if they were homozygous recessive. Phenotypically, these two are the same. Okay? Phenotypically, these two are the same, but we won't worry about him right now. So we can make a quick little Punnett square to determine the possibilities of different genotypes and phenotypes were these two individuals to have offspring. So what I've done is I've taken the two alleles and I've written them on the outside because these are the possible gametes. These are possible gametes. What I'm writing here is that this says that there's a possibility that with this genotype that there are gametes that have the big R and they have the little r. After they go through this process, and all their chromosomes, their chromosome 1A and their 1B gets duplicated and then split, that this guy could end up being the big R or the little r. And perhaps then this guy is the opposite. So again, these are the possible genotypes of the gametes. These are possible genotypes of gametes. Possible genotypes of gametes. All right. And at this point in time, now we can do our genetic multiplication, basically. And what I mean by that is we take our one variable here, r, and we can multiply it by this r. And so we have r squared, or we might write it out as rr. And so I'll do the same thing here. Big R times a little r might end up being big R, little r. Little r times big R would end up being little r, big R, or you could write big R, little r. Both of these are completely okay. If you wrote little r, then big R, or big R, then little r, that's completely okay. Both of these tell you the same thing. They're just written in two different ways. Again, I've mentioned this in class, but I'll say it again. You'll generally see it written more dominant first, recessive second, but both of these are viable responses. And last but not least, we have our homozygous recessive, little r, little r. And so now we can take a look and we can say a couple of things. The first thing we can do is we can write a genotypic ratio and a phenotypic ratio. So when we're talking about the genotypic ratio, that means we're looking at what is the ratio of the genotypes, or what are the possibilities, what are the frequency of the possibilities of the genotypes. Well, I see three different genotypes here in this Punnett square. I see one homozygous recessive. I see two heterozygotes. And I see one homozygous. I'm sorry, I might have said homozygous recessive here, but I meant dominant. When I wrote this first one, this might have been recessive, but I meant dominant because this is now our homozygous recessive. So that is our genotypic ratio. You could also write it as like a 25% chance of homozygous dominant, a 50% chance of heterozygous, or a 25% chance of homozygous recessive. That is also completely okay. If you wrote in the numerical ratio like this, I would encourage you to define what each of these numbers are representing. Otherwise, you're just writing numbers that have no meaning. Now that we wrote our genotypic ratio, now we can do our phenotypic ratio, and that's not supposed to be ration, it's supposed to be ratio. So now what we have to do is you have to take a look at what our genotypes are telling us. Well, real quick, let's go back and look at the information we've been given. Again, don't forget that we've been given that the low heart rate is dominant to the high heart rate. So that means that anytime you see at least one big R, that means that that individual who has this possibility of this genotype will show the dominant trait, or in this case, the low heart rate. And so I see one, I see two, and I see three individuals that show the dominant phenotype, or the low heart rate. And I see one individual that shows the recessive, or the high heart rate. Again, you could also write maybe something like it's 75% here, and it's 25% here. Either one of these is fine. Again, just as long as you make sure that you clarify and you define what these numbers actually mean. So that's all nice and easy, but what if we're looking at two genes at the same exact time? What if we want to look at if an individual is going to be, what they might have when it comes to heart rate, or we can look at cystic fibrosis. Cystic fibrosis in humans, if you missed it, is a disorder where your respiratory system and your digestive system don't work properly. You produce a lot of mucus in your lungs. You don't have the proper ability to digest some salts, and so you actually might taste a little salty on the skin. And so it's something that is fairly life-threatening, but is also fairly manageable at the same time. So with that, we can go ahead and say, well, what if we've got an individual who is heterozygous for heart rate? I'm going to continue using our heart rate example. They're heterozygous for heart rate, and they're heterozygous for cystic fibrosis, in that they do not have it. Note that the recessive trait is cystic fibrosis. The dominant trait is not having it. The recessive is having it. And we will cross this individual again with someone of the same genotype. So now we have someone who has a low heart rate with no CF, and they're mating with someone who also is low heart rate with no CF. Generally, sounds like a fairly healthy individual so far. Just like we did here, we need to find out the actual possible genotypes of our gametes. However, it's not as simple as it was this time. Now we've got another variable in play here, so we need to account for all possibilities. So this is where we do our biological foiling. So that means I'm going to take this first allele of the first gene, and I'm going to potentially match it with the first allele of the second, or the second allele of the second. And so that means that I'll have the potential for a big R, big F, or a big R, little f. And I'll do the same thing with the other allele for heart rate. So that I have little r, big F, and little r, little f. This tells us that these are the 4 possible alleles for this individual. Should their cells go through meiosis and they produce sperm cells or egg cells, looking at these 2 traits, these are the 4 possible combinations that they could get matched together as. And because we're looking at someone of the same genotype, it's going to be the same for this person as well. So again, I match the first with the first. I match the first with the second. I match the second with the first. And I match the second with the second. And now we can set up our Punnett square. However, this time, where this first one, our monohybrid, was a 2x2, we're actually going to double everything. When I say double, I don't mean 2 becomes, or 4 becomes 8, but I mean that 8 or 2 across this way will become 4 this way, and 2 this way will become 2 down. And so this becomes a 4x4 for 16 total squares. For 16 total squares. And now, in the same way that I took my gametes, one second, I'm sorry. In the same way that I took my gametes as possibilities from here, and I split them up, I'll write them up here in the same way. So big R, big F is this one, and then I'll match them all the way across. So big R, little f, little r, big F, little r, little f, and that is this individual. And so then I'll do the same thing here, but we know that they're actually the same. And so now we can do our genetic multiplication again. So in the same way where I matched over here, big R to the little r, so I got big R, little r, we'll do the same thing. So I get big R, big R, big F, big F, big R, big R, big F, little f, big R, little r, big F, big F. And I just pull that all the way across, again, matching here. If you wrote something along the lines of this, little f, big F, again, don't forget that that is completely okay. Because this is still genetically the same thing, that is all good. However, the one thing we'll say is do not do something like this. Do not mix the letters between each other. Make sure when you do this that you put the like terms with each other. So this should actually be written as big R, big F, little f. Don't write just big R, little f, big R, little f. In fact, I actually did mess that one up. So again, I've got big R, big F, little f, and this is a big R, little f, little f. So I'm just going to go ahead and real quick finish this up. Okay. Now that you've completed your Punnett square, now again, we can set up our phenotypic ratio and our genotypic ratio. I'll be honest with you, this one is not going to be fun. I'll tell you that right now. So we'll do this one first. And the reason this is not going to be fun is because, well, there's a lot of different genotypes going on here. So again, if we're looking at genotypes, I see only one that is homozygous dominant, homozygous dominant. There's one homozygous dominant, homozygous dominant. But now here's a homozygous dominant, here's a heterozygous homozygous dominant. And so I'll be honest, we're not even going to do this. If I really wanted to be a jerk, I would make you do this. However, I don't want to do this, and so I'm not going to make you do this. So in a dihybrid, we'll just focus more on our phenotypic ratio. Don't worry about your genotypic ratio of the dihybrid as much. If you want to practice it so you can practice your ability of recognizing the genotypes, more power to you. Feel free. But it's a pain in the butt, and I recognize that, so I'm not going to just outright do that. So for our phenotypic ratio here, remember that our phenotypic ratio is looking at the actual traits. What are the traits showing here? Let me refocus this. Looks like it's starting to unfocus. There we go, a little bit better. Let me walk back here and point something out to you. We originally made it a true heterozygous with a true heterozygous. We're actually going to get a really, really nice looking pattern here. And so in the end, I'll show you that pattern again, but be ready for that. So phenotypically, we've got two traits to look at. We've got, are they high or low heart rate, or in this case, this individual's low heart rate, and do they have cystic fibrosis or do they not? So right now, I'm just looking at the dominant. So I'm looking at, do they have a low heart rate? Do they not have cystic fibrosis? So that's this guy, right? At least one dominant allele, So that's this guy. In fact, it's also this one, and this one, and this one, and this one, and that one, and that one, and that one, and that one. When you set up a true heterozygous like this, in the same way that I have, you'll actually find that this will be part of a visual pattern where the dominance will be all the way across the top, all the way down the bottom, and then finishing the diagonal here to connect them. We can just make sure. So I see a dominant trait here, dominant there, dominant. Good. So that is 1, 2, 3, 4, 5, 6, 7, 8, 9 that are dominant. Now let's do my dominant recessives. I'm going to do my dominant recessives. Notice that, again, I am clarifying what my numbers mean. So dominant recessives, again, it means that we're looking for guys that have at least one dominant allele in their first gene or their first trait, but no dominant alleles in their second. So that would be this one. That would be this one and this one, and that's it. I see a big R here, a big R there, a big R there, both little f's, both little f's, both little f's. So I've got 3 that are dominant recessive. Now I'm going to do my recessive dominance. So that means that we're looking for no big R's and at least one big F. And so I see 1, 2, 3. Little r, big F, little r, big F, little f. So there are 3 that are recessive dominant, and then there is going to be 1 left that is recessive. So what this means is that there are 9 possible individuals, there are 9 possible phenotypes, 9 possible genotypes that all create the same phenotype of low heart rate and no cystic fibrosis. There are 3 possible genotypes that tell us that we'll have the low heart rate, but with cystic fibrosis. There are 3 possible phenotypes that tell us that we'll have the high heart rate, but no cystic fibrosis. And then there is only one genotype that tells us we'll have both high heart rate and cystic fibrosis. Now, again, you could write these out as percentages, but this is actually saying that there is, 9 out of 16 here, and there's 3 out of 16 and 3 out of 16. So like if you wanted to do 1 out of 16, go for it, but it's kind of a waste of everyone's time. Then you got to pull out a calculator. So if you just wrote like a 9 to 3 to 3 to 1 like that, as long as you clarify what this means, what does the 9 mean? What does the 3? What does this 3 mean? In fact, I would encourage you to actually say that this, instead of being dominant recessive, that this 3 is low heart rate with CF, and that this 3 is high heart rate and without CF. I would encourage you to clarify that a little bit further like that. But as long as you clarify at least a little bit what we're looking at here, then I'm happy. So that's that. I hope that this was somewhat helpful. Again, I strongly encourage you to see if you can't find someone else, Amoeba Sisters, Khan Academy, anyone to walk you through this as well. It's often so much better to to hear other people explain it to you, not just the same nasally voice over and over and over. I also encourage you to both this and some of the other topics for this unit. Try teaching your friends, try teaching your family. Especially something like genetics kind of gets the whole family involved, and it's kind of neat for everyone, where you can trace some of that stuff back a little bit more. So I highly encourage you to use the material, not just learn the material. But with that, that's the end of this series. Again, good luck this week. See you Wednesday, see you Thursday, and after that, have a wonderful break.